Genes are the building blocks of life and contain the information that is needed to create proteins which determine the characteristics of an organism. The Human Genome Project, as described by the National Human Genome Research Institute, is the international collaborative research program whose goal was to complete the mapping and understanding of all genes in human beings. The HGP is dependent on the preliminary findings of early genetic scientists. This includes the creation of the first gene map in 1911 when Sturtevant discovered the loci of the Drosophila or fruit fly gene for eye colour. Equally important was the discovery by Watson and Crick of the double helix structure of DNA molecule in 1953. Techniques for sequencing DNA were first developed in the 70s by Frederick Sanger, who brought about the idea of sequencing the human genome in the 80s. Emphasis in the years prior to the undertaking of the project was focused on the development of technologies to accelerate and ensure the accuracy of findings. Various techniques were used and invented to make the Human Genome Project possible. This includes techniques to create large quantities of identical DNA, ways of cutting specific pieces of DNA, and ways to separate DNA strands according to length. Restriction fragment polymorphism is used to break DNA into two pieces at a specific site using restriction enzymes. Yeast artificial chromosomes are artificially created DNA molecules that are used to clone the DNA inserted into yeast, which creates a large volume of the desired DNA. This process is vitally important as sequencing is not possible with large quantities of genetic material. Polymerase chain reaction also creates large volumes of genetic material. The technique involves the denaturing of DNA, which releases the hydrogen bonds and separates the two strands. Polymerase is then added, which attaches to a primer and begins to create the complementary DNA strand. These processes are then repeated to create an exponential volume of genetic material. Electrophoresis is used to identify varying lengths of DNA. DNA samples are placed in indentations in the gel slab. The gel is then charged with an electric current. As the DNA is negatively charged, it will be attracted to the positive end and will move through the gel. This separates the DNA strands according to length. Recombinant DNA was first produced by Paul Berg in 1972 and was the first technique that enabled the large-scale replication of genetic material. The portion of DNA would be identified and cut by restriction enzymes, and this piece of foreign genetic material is then placed inside a plasmid in yeast, bacteria, or a virus to form a circle. Each end must have complementary base pairs and must be sealed with enzymes. This plasmid is then transcribed and translated by the organism in the same way as if it was its own. This technique was replaced by yeast and bacteria artificial chromosomes and later by polymerase chain reactions. The HGP has resulted in improved diagnosis of disease. Doctors are able to use genetic tests to identify common genetic disorders by identifying similar variants in their genetic code. Identifiable diseases include leukemia, hemophilia, Parkinson's disease, and Williamson's disease. Genetic testing can confirm a doctor's initial diagnosis and allow for treatment to be implemented earlier. The HGP has allowed for earlier detection of genetic predispositions to disease. Newborn screening is offered to all babies born in Australia. According to Better Health 2016, at 2 days old the baby is pricked on its heel and the blood is tested for genetic disorders such as PKU, hypothyroidism, and cystic fibrosis. Earlier detection of these genetic diseases means that measures can be taken to minimize the effects of this disorder on the child, and the child can be properly cared for, taking into account its special needs. Drugs in the past have been created and prescribed using trial and error, but advances in genetic understanding of diseases have resulted in rational drug design. According to the Pharmaceutical Journal, this technique, rather than using trial and error, identifies the target mutant gene and tailors the drug to that specific mutation. This has allowed for the discovery of drugs to become a more efficient process. The pharmacogenomics technique also identifies what drugs will be effective in specific individuals and what drugs will not be, thus resulting in faster, more effective treatment of patients. Gene therapy is an innovative way of treating diseases and is only possible through genetic sequencing. This technique involves the manipulation or insertion of genes into body cells. The culprit gene can be turned off, replaced, or they can insert a new gene to re-establish normal gene function. It is hoped that in the future doctors will be able to treat diseases by inserting genes into cells instead of using drugs or surgery. The HGP has contributed to many benefits, but it does not mean scientists know everything about genetics and disease. There remain many limitations to the findings of the HGP. This includes genetic diseases that are caused by mutations in the non-coding regions of DNA called introns that regulate gene activity. These genes, previously known as junk DNA, do not code for proteins and are not as thoroughly understood as protein coding regions known as exons. Many mutations that cause disease are not the result of a single gene, but an array of genes. This is known as polygenetic inheritance. These diseases are much more complex and harder to detect. For example, the identified genes that cause breast cancer only accounts for 10% of all breast cancer cases. Many diseases remain much too complex to be detected and treated through genetics. The HGP sequences all the genes in the human body, but just sequencing one gene does not tell us its function. Scientists are able to figure out the sequence and therefore the amino acid chain, but they do not always know how the amino acid chain will be folded in the rough endoplasmic reticulum to create a protein. Scientists do not know the functions of approximately 50% of genes, so in those cases the HGP is not useful until further research is completed. Linked genes sit close together on a chromosome, making them likely to be inherited together. Genes on the same chromosome are linked, while genes on separate chromosomes are unlinked. The degree of linkage determines the likelihood of certain characteristics being inherited together. Genetic linkage maps are created by scientists to find the relative locations of marker genes on chromosomes and the distances between those genes. These maps are useful when studying organisms who have not yet had their genes sequenced, for example plants. Genetic linkage maps provide a basic understanding of an organism's chromosomes and is useful for checking the accuracy of genome sequencing. Here is a chromosome that contains the genes A, B, C, and D. To determine the location of these genes, experimental crosses must be undertaken. If in the resulting cross, the characteristics for A and B are expressed together 90% of the time, those genes are located approximately 10 units apart. This process is repeated for the other genes using the same technique. It is known that genes A and B are located 10 units apart, but it is not known which is located above or below the other. The genes A and B must be compared to that of C or D to confirm their location. If A is inherited with C 55% of the time, then those genes are located 45 units apart. If B is inherited with C 65% of the time, then they are located 35 units apart. Therefore, it is logical that gene A will be located above gene B because the distance to C from A is longer. Using linkage maps to map the human genome would be almost certainly impossible. The long lifespans of humans and high gene maturity means that experimentation would take too long and would be considered unethical. Linkage maps are useful to locate the genes of organisms with short life and reproductive cycles, for example rabbits and flies. As linkage maps are reliant on the testing of more than one generation, these organisms are useful as the period between generations is short and results can be gained at a faster rate. Secondly, linkage maps are only able to locate genes that result in a characteristic expressed through the phenotype, for example eye and hair color. Genes that code for internal changes would not be visible and cannot be tested. Linkage maps only locate marker genes within the chromosome, but they do not locate every gene. The HGP mapped every gene in the human body, and that is not possible through linkage maps. Genetic linking is highly reliant on the averages of test results obtained through experimentation, though these results are not certain. The rates of recombination give an indication of the distances between genes, but this information is only an approximation of the distances. This means that the results obtained from linkage maps cannot be relied upon to be 100% accurate.